thanks again for putting this workshop together in these challenging times. I just want to lay some groundwork on this, and I really want to thank Matt Keir for his synopsis of what the Plant Extinction Prevention Program deals with as far as number of species and number of days we have for each species. And as sobering as that was to see, I think it was a little bit optimistic because we don't spend the 2,000 hours a year of our employment in the field. We have data entry to do, GPS, GIS work to do. There's trainings that the university requires of us to do. We have to pack for camping gears. We got to unpack and decontaminate gear, et cetera, et cetera. So we don't come near to doing 2,000 hours of field work a year. And the other side of that is he just said, well, if you work with this species, that's how many days. But some of these species are really spread out. We don't just go to one place and we get to work with all the individuals or populations. Some of these species are in several populations, really spread out, that require helicopter time. So although we might be technically working with that species, we're not necessarily working with all the plants in that species. So it's much less time per species than Matt's great slide alluded to. And the reason I bring that up is, as resource managers, you know, we do rely on the scientific and academic community to answer some of our questions and give us guidance. Because we really don't have the time to spend on plants that maybe aren't a good species, or we're ignoring things that are slipping through the cracks. And, you know, we have a real sense of urgency as these things decline. So I just wanted to, you know, put that out there as a basis for what my talk is about. Next slide, please, Gary. So the first thing I wanted to talk about was hybrids. And next slide, Gary. This is Melanthera camolensis at Kapuni. This is the type location for this rare narrow endemic on the leeward slopes of Haleakala. Next slide, please. This is in an area that has a lot of goats. Next slide, please. And in an effort to save the last few plants with the Leeward Haleakala Watershed Restoration Project, we built a fence. Next slide, please. But there was a large wildfire, and that, if it didn't kill the plants outright, it melted the fence and the goats got in again. And now all the plants, as far as we know, in the type location are extirpated, although we do have material ex situ. Next slide, please. However, about 4,000 plus meters away in Alena, there seems to be another population of this species. Next slide, please. However, there's also hybrids with Lipokita rockii, or I don't even know what to call these anymore, because I heard they were all going back to Lipokita. What's interesting about this, there's also some sandalwood hybrids in this area with the common Santalum ellipticum and the endangered Lanai sandalwood. So that's kind of interesting. Next slide, please. And you can see the difference in leaf morphology with Melanthera camolensis on the left and Lipokita rockii on the right. Next slide, please. So we've recently retrofitted an old exclosure to protect these plants. But we're devoting a lot of time to things like fixing the fence, weed control, trying to collect seeds, but we're not sure if any of these plants in this other location are, what we would call, quote unquote, pure Melanthera camelensis. And we did collect some seeds from a few plants several years ago that we got some seedlings back from Lion Arboretum, the seed conservation lab, from their germination trials, and one was clearly a hybrid, but the seed wasn't collected from one of the hybrid individuals. And so, the question becomes, was that a recent hybridization event, or was there something buried in this plant that looked like the good stuff that was expressed in the seed? And so it's really confusing. And then the question is, and I think we'll be talking about this at the end and throughout the day, is, what do we do about the hybrids? Should we kill all the hybrids? Should we remove the parent, the common parent, et cetera, et cetera? Next slide, please. Okay, so I think we'll deal with all the questions later. Next slide, please. Another issue with hybridization is this Peperomia subpetiolata, which is also a narrow endemic on the windward slopes of Haleakala. This is allegedly a hybrid based on its pubescent stem, although I think it's what Juncker had described as Peperomia culensis. Next slide, please. This is looks more like the real thing. It's more glabrous. Next slide, please. And this is a volunteer from some of the outplantings that we have done. And one of the lucky ones that the slugs didn't eat. Next slide, please. And they also readily volunteer at the Olinda Rare Plant Facility under the benches. And so we dig these up and we do plant them. And I've been told different things from different people. One is plant them all. And other people say you want to select the offspring to be closer to the pure Peperomia subpetiolata. And there's two other parents that have been that are allegedly parents. One is Peperomia cookiana, which does occur in these hybrid swarms, and the other is Peperomia hirtopetiolata, which seems to occur further east in wetter forests, but their ranges might have overlapped at some point. Next slide, please. And this is just a weird thing in the nursery, how the spikes are resuming vegetative growth. Next slide, please. But the seeds are viable, and that's one of the things that supposedly point to something not being a hybrid is, the seeds are good, the pollen has high stainability, it doesn't resemble other parents, et cetera, et cetera. But these do make lots of viable seed, which we outplant. Next slide, please. And so I think we'll just deal with the questions at the end. Next slide, please, Gary. And then the issue that I think we're looking to genetic analysis to help solve is new species. And next slide, please. Although this is not a new species, this is Sertandra kipuhuluensis. This is something that was described by Harold St. John many years ago, and the manual, and not to be contrary to Warren, but the manual has it as a Sertandra spathulata Sertandra paludosa hybrid. And, you know, we have to remember the manual, the cutoff date for the manual was in 1987. That was almost 35 years ago. And so there's been a lot more field work and a lot more collections. And I think we know a lot more about a lot of species than we did back then. And this species seems to be not just in Kipahulu, but across all of windward Haleakala. It's very distinctive. Next slide, please. Has these really prominent square stems, which we affectionately call SpongeBob square stems. Next slide, please. And really distinct zygomorphic floral morphology with the 3 dorsal calyx lobes kind of fused and the 2 ventral ones split to the middle. Next slide, please. And then there's the issue of if something is a rare and endemic taxa or it's indigenous and widespread. And here's a couple examples. Next slide, please. This is Cyperus odoratus, or what we think is or was Cyperus odoratus. Unfortunately, this small population that K.I. Bustamante found is now been destroyed when the, well, I shouldn't point any fingers, but yeah, it's been destroyed. We have plenty of seeds in storage and in the nursery at Maui Botanical Garden, but it may be a widespread indigenous species, but it might be a Hawaiian endemic. And I think Warren is working with somebody, I think it's in Mexico, that is hopefully going to help us solve this issue. But it would be a loss to lose the Hawaiian populations of it, but it would be a real tragedy if the species went extinct, if it is a really narrow endemic. Next slide, please. And the other one that's been a mystery is this Lepicinia hustata, or pakaha. That's in the Lamiaceae. Hillebrand considered it a native species, widespread on Haleakala. Now it's only in a few places, and we don't, you know, we again, should we be working with it? I know there's projects that have outplanted it. I know I have outplanted in a couple of places when time allowed, but you know, is it a native species? Next slide, please. It seems to be really widespread in Mexico. I don't think, you know, we need to worry about the species going extinct, but again, it would be a loss to see the Maui/Hawaiian population be extirpated. So, you know, we need some maybe genetic work to find out, like, what is the status of this? Is it introduction? Has a really interesting story that, you know, it might possibly have been introduced by the Spanish a few hundred years before Captain Cook arrived in Hawaii. So next slide, please. And here, just some more questions. This is the end of the slide. Maybe this is a good time to begin the discussion. I think one of the possible silver linings of COVID-19 is these rapid tests that have been developed. And maybe this technology could be applied to plants in the field. I don't think we're expecting to be able to have such a portable kit and give us an answer, you know, immediately in the field, but maybe, you know, taking it back to your base yard or camp and getting an analysis in a couple of hours or a couple of days. Right now, it takes years before we get an answer the way we are dealing with things. And we just don't have the time for this. These things are declining rapidly from 1 or 2 small populations. And I'm hoping there's some breakthroughs in genetic analysis. Angela from Chicago Botanical Garden gave us a great overview of all the different methods and things. And I'm hoping at least one of those is field and user-friendly. The other thing is, how do we identify some of the nearest congeners to avoid hybridization in our reintroductions? And how can we breed out the quote unquote undesirable common parentage and keep the good rare stuff? I know I've discussed, and this is, I'm looking at you, Sir Tander, but I don't want to name any species or locations, but there is a really rare Sir that is hybridizing with a really common one. And I kind of inquired about, can we kill all the hybrids? Can we kill the common one? You know, it's genetic pollution. And the answer was, and this is true, like speciation is sometimes driven by hybridization. But in this day and age, I'm thinking hybridization is also driving extinction, is it not? So that's a point of discussion. And then, you know, what type of material is best to collect? If it's in silica, that's easy. If it's fresh, it's logistically challenging. You know, typically if we're camping Monday through Thursday or Friday, we can't get it to the post office in time where it will arrive somewhere, you know, in America or anywhere in the world by Monday and be in good condition. So we have to consider when we're making our collections that we could get to the post office or FedEx. And I think we just defer to our colleagues in the lab as the best types of extraction and analysis for each group. So that's about it. That's all I got. I think that's probably enough. And I really look forward to hear what Josh and Eric have to say and what the group discussion is going to be. So that's it, Gary. I'm done.